Thank you very much. Dear committee, welcome everybody, ladies and gentlemen. Today I would like to show you our recent work and preliminary results, focusing on the investigation of microvascular parasite contractility in age-related ischemic brain pathologies. It is well known that aging is a major risk factor for several neurovascular diseases. Among these, vascular cognitive impairment and dementia, VCID, is the second most common cause of age-related dementia after Alzheimer's disease, accounting for approximately 20% of the cases. VCID is caused by a chronic and significant cerebral hyperperfusion, or ischemia, which basically means a lack of blood flow in the brain. It comes with diverse cerebrovascular pathologies, including atherosclerosis, microinfarcts, microbeads, and white matter changes. In these ischemia-associated conditions, the blood-brain barrier and the neurovascular unit dysfunction actively contributes to the injury pathogenesis. So understanding the mechanisms behind neurovascular unit dysfunction in such pathological conditions may allow us to develop potent and effective therapies for prevention and treatment of neurovascular diseases. But what is the neurovascular unit and why is it interesting for us? The central nervous system and its circulatory system are tightly connected via the neurovascular unit, or MVU for short. The MVU represents a close structural and functional relationship between microvascular endothelial cells, pericytes, glial cells, and neurons. Among cells of the MVU, pericytes are the least well-defined and most controversial cells. Multiple studies have already shown that capillary pericytes can do vessel constriction, but the main question is still how pericyte contraction can regulate age-related microvascular dysfunction. So to address this fundamental question, we use an animal model of microinfarcts in which microocclusions, small vessel blockages, are produced in the microvasculature throughout the brain. With this technique in our hands, we can conveniently identify blocked vessels in ex vivo and in vivo methods, so we are able to study the role of pericytes in the local ischemic response. Our experiments were carried out on young male transgenic reporter mice whose pericytes were labeled with red fluorescent protein. First, we injected 4,000 fluorescent microspheres into their internal carotid artery, while the external carotid artery was transiently blocked. We made 3 different groups, a 30-minute, a 60-minute, and a 120-minute group, based on the time period between injection and the termination of the experiment. Animals were sacrificed and then perfused with paraformaldehyde fixative, and afterward we made 150 micrometer thick coronal brain slices. We took a series of images from these brain slices with a fluorescent microscope, and then we made a 3-dimensional reconstruction of the distribution of microspheres in the brain. We observed that in all groups, a considerable amount of microspheres got lodged in brain microvessels. The majority of microspheres that caused vessel blockage were located in the ipsilateral hemisphere of the brain, which means that on the same side as the injection was carried out. By analyzing the regional distribution of microspheres, we found that at the earliest examined time point, 30 minutes, the occlusions mostly occurred in the neocortex. We saw a quite heterogeneous distribution of microspheres in the longer time period groups, but we found an increase in the number of large microbeads, mostly in the hippocampus. So because we saw an increased number of microbeads in the hippocampus, we started to study the structural and functional changes that are caused by the large microspheres in the mentioned brain region in real time. In this experimental setup, we first implanted an optical cannula above the animal's hippocampus, and after at least a month-long recovery, we injected fluorescent microspheres into their internal carotid artery the same way as I explained it before. After this, 30 minutes after the injection, we imaged their hippocampus with in vivo 2-photon microscopy to visualize the occlusion sites created by the lodged microbeads. So with our method, we were able to visualize functional microvessels and stuck microbeads in the hippocampus of anesthetized mice. On the left, you can see a real-time flow of blood stained with Evans blue dye in microvessels, and on the right, the bright fluorescent spot indicates a stuck microbead in a vessel at 100 micrometers below the surface. So as I mentioned in the beginning, one of the most interesting questions of cerebrovascular research is the role of parasite contractility and the mechanisms controlling this action in both physiological and pathological conditions. For this reason, we made immunofluorescence staining for alpha-smooth muscle actin, a protein that is mostly responsible for vessel diameter changes on microsphere-injected brain sections. What we found was that the alpha-SMA expression was noticeably increased in parasites that were located in the vicinity of microspheres. Moreover, some vessels constricted near them, which implies that the observed vasoconstriction might have been caused by parasites. However, this event did not occur in every case. It might be because microspheres got lodged in smaller microvessels where parasites no longer express alpha-SMA, or there are other contractile proteins that have a role in the contraction of the vessels. So to further address this question, we developed a new protocol for immunofluorescence staining of alpha-SMA, with which we could minimize the depolymerization of actin filaments and find the expression boundary for alpha-SMA in the microvasculature of the brain. What we found was that the fourth-order vascular branches were the expression boundary for alpha-SMA expression, indicating a transition from pre-capillary arterioles to what we call true capillaries. This result is in line with previous research findings and also raises the question of the possible role of contractile proteins other than alpha-SMA in the contraction of parasites in true capillaries. So in conclusion, we can say that the animal model that we use is suitable to study the dysregulation of microvasculature in ischemia, in which parasites might play a crucial role. In our in vivo study, we demonstrated that employment of optical cannula allows us to study microocclusions in deeper brain regions. Based on our immunofluorescence stainings, it appears that parasites that express alpha-SMA can cause vessel contraction, but this contraction doesn't occur in every case. So currently we are doing experiments to find the exact protein that is responsible for parasite contraction in the occlusion of true capillaries. We hope that our current research takes us closer to understand the mechanisms behind pathological conditions of the neurovascular unit and the consequential vascular dementia. First, I would like to thank the National Scientist Academy for still supporting my work at Kriszbai István's laboratory, and I would like to thank István for mentoring me and the whole group for the joint work. Thank you for your attention.
Thank you very much, Zsófia, and we would like to present a certificate for your talk.